Okay, so we talked a little bit about cancer last time, and now we're going to talk a little bit about some genetic-based cancer therapies and aging. First, I wanted to ask you a warm-up, though. What are telomeres? You might remember them from biology. Telomeres are repeat DNA sequences found at the ends of chromosomes. Remember, we don't have circular chromosomes, we have linear ones. And they're functionally linked to the number of cell divisions before a cell undergoes programmed cell death. Now, a couple of genetic therapies. One approach has been to try to use tumor necrosis factor. Now, tumor necrosis factor is produced by white blood cells, and its release kills nearby cells. So there's a special type of white blood cell called the tumor infiltrating lymphocyte that releases tumor necrosis factor. So one therapy is to pull these tumor infiltrating lymphocytes out and in vitro genetically modify them so they produce more tumor necrosis factor. It's like the ones that you were successful, grow them up, and then inject them back into the patient. And you try to help the patient fight the tumor using basically their own cells that have just been enhanced. So these tumor infiltrating lymphocytes, as you can see here with the little masked bandit, are releasing tumor necrosis factor, and that will help kill tumor cells. And that is infiltrating into the tumor from the bloodstream. Here's that ex vivo gene therapy where they were talking about in the previous slide, that you pull the white blood cells out, genetically modify them, select them, make sure that you've got cells overexpressing tumor necrosis factor, and then you inject them back into the person, and then hopefully they will kill the tumor. Another genetic approach has been to transfect tumor cells by a process called lipofection. And they use the genes to human leukocyte antigens. And what you do is all cells express human leukocyte antigens. Each person has a unique set. And our bodies recognize them as ourselves, so our immune system doesn't attack them normally. So what we do is we'll take genes for human leukocyte antigens that have nothing to do with us, that are from some other person, and by lipofection, by putting them into the tumor cells, we will cause them to express human leukocyte antigens that are not self, and then our immune system will destroy them. So lipofection is basically means you have a liposome, which is basically a hollow, sort of like lipid bilayer, little vesicle, and it's got our genes to be delivered, and you inject it directly into the tumor, and it will fuse with membranes and deliver the cargo, which of course the cargo are human leukocyte genes, human leukocyte antigen genes that are not from us. Okay, telomeres. Telomeres are found at the ends of chromosomes. They consist of hundreds of repeats of TTA, GGG. A few are lost every cell division. After about 50 cell divisions, enough are lost that the cell will undergo apoptosis. So apoptosis is programmed cell death. Okay, so these shortening of telomeres has been linked to aging. So as you get older, you're losing some of these repeats from the ends of your telomeres. Now they can be restored by this enzyme called telomerase, but also levels of that decrease with aging. Now in tumor cells, one way they can avoid undergoing apoptosis is by activating the gene telomerase in these tumor cells, so it keeps rebuilding the telomeres so they never stop growing. So one way of perhaps treating individuals with cancer that have this gene overexpressed would be to inhibit it. And so if you can inhibit telomerase, you might be able to kill the cancer cells. Okay, moving on. Things we learned from worms. So the worms that we're going to talk about are C. elegans. C. elegans are flatworms, and they consist of about 959 cells in the adult. They actually have more cells than that, but some of them undergo programmed cell death as during development of the worm. And C. elegans is used or studied to understand how organisms develop. So there's one mutant of C. elegans called DAF2, and DAF2 C. elegans worms live twice as long. So it seems to be a link between this gene and aging. So longevity seems to be linked to metabolism. So mutant DAF2 worms live twice as long, but they metabolize nutrients more slowly. So the question is, Does a lower metabolic rate help you live longer? It may. There is some anecdotal evidence that if you have caloric restriction in a human and you're eating a minimal subsistence diet in terms of calories, you can actually potentially live longer, but it's not a proven study. Here's just a cute little picture showing the flatworms. So you can see at birth, wild type, DAF2, perfectly happy. Day 25, the wild type, little X's in their eyes, they're dead. DAF2 ones, perfectly happy. Day 50, perfectly happy. Meanwhile, the wild type ones are, well, decomposing. And then finally, you get somewhere between day 50 and day 60, the DAF2 mutants will actually die. So apoptosis, like I said, is programmed cell death. And the way it was kind of really discovered and really kind of like at least partially figured out was by studying what the phenotype of the SED9 gene was in the development process of C. elegans. So when SED9 is defective, cells do not undergo apoptosis. So instead of growing up and having 959 cells, they end up with a little over 1,000 cells. And so some of those cells that were supposed to have died don't die, and they end up not being a very healthy little flatworm. So SED9 delivers a signal for cell death to two other genes, SED3 and SED4. So these are all C. elegans genes. And so SED9 sends the signal, and the ones that actually execute the cell, part of our genetic death squad, are SED3 and SED4. Now, humans, we don't have SED9, but we have something called BCL2, and BCL2 seems to function similarly. So cancer cells don't have a functional BCL2 gene, or many of them don't. So in other words, if you have a cancer cell and it's got a functional BCL2 gene, it will potentially undergo apoptosis. But if you don't have a functional BCL2 gene, they can just keep growing and growing. It doesn't matter what happens to the telomeres. So cancer cells usually have this mutant form, and they can't be destroyed by apoptosis. And that sort of is one of the factors that contributes to their ability to continue growing. Last thing I want to talk about is mitochondria. So mitochondria are the energy producers for the cells. You inherit them from your mother. They come from the egg. Energy production decreases with age due to mutations in the mitochondrial DNA. If you start out with defective mitochondria, your life expectancy is shorter. So there you can blame your mom, because all the mitochondria that you you begin with, come from your mother. And so if they're defective right off the bat, your life expectancy is generally shorter. Like I said, mutations tend to accumulate. If you look at human mitochondria DNA, so as cells grow and divide, the DNA inside the mitochondria grows and divides, and you end up with mutations happening, and a common one is a 5 kilobase deletion. That region removes several energy-producing genes that are important for proper mitochondria function. So you end up not having as much energy, and that's not a good thing, because that actually, if you lose too much energy, you don't live longer, you die.